So let me just start by thanking the organizers, specifically Abhishek, for this kind invitation to present our work at this nice meeting. I've been following this meeting via YouTube actually since morning. Um, I also thank all the participants and everybody on the YouTube who has stayed late to hear me today. So the idea of today's talk is to basically present two pieces of work which I have done in my group. Uh, we do several things, but I would just like to present two stories today. Uh, I would try to present the first story as much as possible completely, and the second story, if I have time, I would try to present it as much as possible. So with that, I'll talk about some chemical spaces, and there is no machine learning or anything which is going to be in my talk, but we'll talk about some data curation and generation and analysis of highly curated data sets which are relevant for biology. So specifically, I would present in the first part of the talk on a chemical space, which is the phytochemical space of Indian medicinal plants, which is potentially beneficial to humankind. And then the second part of the talk, I don't know how much time will permit, I will talk about our work on a potentially harmful chemical space for humankind, which is the space of endocrine disrupting chemicals. So with that, let me just first go to the first part of the talk. As you know, plants use phytochemicals as chemical defense mechanisms against their competitors. We also know that plant extracts containing phytochemicals have been used to treat various human ailments for centuries. Also, many present-day drugs were initially isolated from plants. Prominent examples include the anti-malarial drug artemisinin and the anti-cancer drug paclitaxel. So, as we are aware, various systems of traditional medicine, including Indian and Chinese, use several of these medicinal plants in their formulations. So, if you look at traditional Indian medicine, they use several plants in their formulations to treat various ailments, but such information, including the information on the phytochemicals, which is the centerpiece of this work, is largely buried within books. So when we started this work, I argued that the non-digital nature of this information on the phytoconstituents of Indian medicinal plants limits its effective use towards modern drug discovery. I would also like to point out that in contrast to Indian herbs, there were many more comprehensive digital databases such as TCM-ID and TCM-Mesh, which existed on the phytochemicals of Chinese herbs. So in short, what we did is that this is a work which we published in 2018, which took about a couple of years to accomplish. We have built the largest digital database, IMPACT, which spans 1742 Indian medicinal plants, 9596 phytochemicals, and almost 1100 plus therapeutic uses. And we expect this platform and the database to facilitate computational approaches towards traditional knowledge-based drug discovery. IMPACT is accessible using any of the three domains which are mentioned below. I would take only 2 slides to walk you through the tedious 2-year process of generating the data set that we have as a database now in IMPACT. So the first step was to get a curated list of Indian medicinal plants. This is not simple because the plant names in the literature are quite synonymous. So we gathered a list of Indian medicinal plants from reports and books from Ministry of Ayush, CMAP, the CSR organizations reports, FRLHT organization in Bangalore, and several other resources. And we tried to build first a non-redundant list of Indian medicinal plants. And for this work, when we started, we had about roughly 5,000 Indian medicinal plants names for which we could search for phytochemicals. So this seems tedious, but this was not that difficult. The step 2 was the main step, which is we wanted to gather the information on the phytoconstituents of Indian medicinal plants. So without going into the detail, the tedious process involved sourcing the data from 3 sources. One, the books, which is highly non-digital in nature, so we had to digitize them. Second, through natural language processing, so we did text mining of PubMed. Specifically, the data was collected from more than 7,000 abstracts, and there were few existing databases in this area. So I should mention that my colleague Ganesh Baglar, who is now at IIIT Delhi, he had built a database of 963 phytochemicals for only 5 Indian medicinal plants called Phytochemica that existed before our work. And there was another work by Polur et al., which captured 1829 phytochemicals of 295 Ayurvedic Indian medicinal plants. Unfortunately, Polur et al. never published a database, and the work did not contain any supplementary material. So we had to extract this information out from the NutriChem database, which claimed to have collected the data from Pallur et al. So as you see, before our work, it was basically limited to around 1,000 or 2,000 chemicals, phytochemicals, and roughly less than 300 plants. So, however, one of the important, uh, you know, centerpiece of our work is that we wanted to have chemicals or phytochemicals in our database with structures only, not just names. And when you go to the books or any resources, the chemical names or the phytochemical names are reported in a very synonymous fashion. So we did a very manually tedious job of removing this redundancy. That is, take the names which are reported in the literature and map them to standard chemical identifiers, including stereochemistry, etc. So, of course, if we found a name which could not be mapped to a chemical identifier, we don't report it on database. To give you an example, Phytochemica lists 963 phytochemicals, but only half of them, or slightly less than that, have a structure associated with it. So in our database, Phytochemica's data will be only half of it. So in this process, what we were able to do in our first round, which is available openly, is some 15,000 common names, which were mapped to 9596 standard chemical identifiers. So essentially, we have in our data space information for more than 1,700 Indian medicinal plants, which involves phytoconstituents that are 9596 in number. So once you have these 9596 phytochemicals with their 2D structures available, you can do a lot of cheminformatics with it. You can gather the 2D and 3D structures. You can compute a lot of physicochemical properties. You can compute the drug likeness. You can predict the admit properties. You can also predict human target proteins using various resources. And all that information was gathered and put into our database. Furthermore, in addition to the information on the phytoconstituents of plants, we also focused on gathering the therapeutic uses of these Indian medicinal plants. As you know, the ethnobotanical or ethnopharmacology of these Indian medicinal plants is a controversial subject. So we didn't do any text mining here. We only resorted to gathering information from very standard books from Ayush Ministry. Again, this was collected from books which are recommended by Ayush Ministry, and we did some minor innovation here as well. Again, the reported therapeutic use can be very synonymous, so we wanted to remove the redundancy, and we mapped the therapeutic uses to disease ontology, mesh terms, and OMIM terms. So we finally have a non-redundant set of therapeutic uses for these plants as well. Finally, on the pilot scale, we also have data on the usage of these plants in different traditional medicinal formulations in Ayurveda, Siddha, etc. Here, we didn't want to reinvent the wheel. There is a large project by CSIR called the Traditional Knowledge Digital Library, where they have claimed to have digitized more than 2 lakh formulations, but only 1,200 of their formulations are openly available. So we just use these 1,200 formulations from TKDL and link them to our plants if the formulations which are openly available in TKDL use any of the plants in our database. Again, we didn't want to reinvent the wheel, so we didn't link to formulations. And our primary interest was to gather the information on the phytochemicals of Indian medicinal plants. So what we have in our database finally is more than 1,700 Indian medicinal plants, more than 9,500 phytoconstituents, more than 27,000 plant phytochemical associations, more than 1,100 therapeutic uses, more than 11,500 plant therapeutic use associations, 974 open formulations from TKDL, more than 5,000 plant formulation associations, and a number of phytochemical human protein associations from Stitch. So, okay, we created this data set, and what can we learn from this? This is just not a database paper, but we have extensively analyzed the data subsequently. The first question I asked is that what could be the potential drug-like space within impact? So I am not a cheminformatician, but my student is trained in cheminformatics. So I asked, right, let's take the wisdom of crowds approach. Let's put in several filters which are used in initial drug discovery pipeline to figure out which are potentially drug-like. So we use 6 different filters. Some of them are used by even pharma companies. And I asked, which are the molecules which pass all the filters that we have evaluated? So we found a set of 960 phytochemicals, which is roughly 10% of the phytochemicals in our database, to be potentially drug-like. What is interesting is that only 32 of these 960 are already FDA approved drugs. And if you ask if any of these 960 phytochemicals have significant similarity, and the similarity is sort of evaluated based on the Tanimoto coefficient, which is a metric used to compute chemical similarity. Uh, so we use a similarity greater than or equal to 0.5 with any FDA approved drug to say that if a phytochemical, which is drug-like, is similar to FDA approved drug or not. We find that only 40% roughly of these 960 have any structural similarity to any FDA approved drug. So another way to look at this drug-like space within impact would be to view it as a network. In this picture, each dot or each node in the network is actually a chemical, one of the 960. And we connect two chemicals if they are similar based on Tanimoto coefficient with a similarity greater than 0.5. So they are structurally similar. The color coding is as follows. A red phytochemical is one, which is similar to any FDA approved drug. A green node is one, which is not similar to any of the FDA approved drug. So you can see there are some disconnected clusters. So the space is highly chemically diverse. There are some clusters with many reds. So they have similarity to FDA approved drugs, but there are also clusters which are purely green. So these are maybe potentially new diverse chemical spaces to explore. We are in subsequently analyzing this space and in a much more thorough fashion, and we will be coming out with more work on this analysis. A second question which is obvious is, and people asked, is that how does our chemical space compare with the chemical space of Chinese herbs? So for that, we took a TCM mesh database because, as you know, to do the comparison, you also need to map the chemical to structures to figure out which are the common chemicals and which are the ones which differ. So we took the TCM mesh database. Luckily, they had given some mapping to the chemicals to identifiers. So it again had roughly 10,000 plus phytochemicals covering more than 6,000 Chinese herbs. Uh, they had not done and evaluated the drug-like subspace. So we did the analysis again using the same filters, wisdom of crowds, and we found that roughly again 10%, slightly less than 10% of the phytochemicals passed all the drug-like filters. So, so again, of the 972 in this database, which passed all the drug-like filters, we found that only 19 were FDA approved drugs already. And if we compare the complete space of 10,000 chemicals plus chemicals here with our impact, we found that there is only a 25% overlap between the two, the phytochemicals in our database and the phytochemicals in the database on Chinese herbs. And similarly, if you compare also the drug-like space, again, the overlap is roughly 25%. So in summary, we find that the phytochemical space of both Chinese and Indian herbs provide extensive opportunity for drug discovery. The final analysis, I don't have time to go in detail, is basically a very involved analysis, but I request you to consult our paper. But I'll try to give you the main message through it because of the lack of time. So this is inspired by work by Stuart Schreiber, who is one of the main leaders of the Broad Institute's drug discovery program. What they have done in a back-to-back PNAS paper is analyze physicochemical properties of three compound sets they have collected. One is a set of commercial compounds, roughly 6,000 molecules, which are used again by pharma industry in the initial drug discovery pipeline. A second set, which is the diversity-oriented synthesis compounds, again roughly 6,000 compounds, and a selective set of natural products, which is roughly 2,500 molecules. For each of these molecules in their sets, they have computed two measures. One, which is known as stereochemical complexity, which is basically the fraction of carbon atoms in any molecule which is stereogenic. Second, which is the shape complexity, which is the fraction of carbon atoms that is sp3 hybridized. Note that these two measures are size independent. So they are defined in a way that they don't depend on the size of the molecules. So what they did is that they looked at the distribution of stereochemical complexity and shape complexity for these three sets: commercial compounds, diversity-oriented synthesis compounds, and natural products. And what they found is that both the stereochemical complexity and the shape complexity is higher in general for natural products in comparison to commercial compounds or diversity-oriented synthesis compounds. But they didn't end here, but they went ahead and did something very interesting. They tested each of these compounds with respect to a random assay of 100 proteins to check how many proteins can these compounds bind to typically. And what they find, and I'll try to summarize in one line, is that they find that natural products typically are very specific binders to protein targets. That is, they find that a natural product will bind to only one protein or two at best, but commercial compounds and diversity-oriented synthesis compounds can be promiscuous and can bind to many proteins. What this means is that if you design a drug molecule potentially around a natural product, it's going to be likely to be highly specific, and it won't have side effects because it will bind to the intended target, but will not bind to other targets which may lead to side effects. So what we did was that we basically cannot do the experiments, but we tried to compare these distribution of these measures for our library, the Chinese library of phytochemicals, and compared it with the the result of Schwartz-Schreiber. So basically, I took these plots and plotted them as a box plot here as a reproduction. As you can see, both stereochemical complexity and the shape complexity of the natural products in our database, which have hardly any overlap with the natural products analyzed by them, is very similar. And if I was allowed to extrapolate based on the results by Schwartz-Schreiber's group, this may suggest that the phytochemicals in both IMPAT and also for the Chinese herbs are potentially likely to be also very specific, in their targeting of proteins and unlikely to be promiscuous. So this may have some advantage if you go for these phytochemicals to design drugs, lead molecules. So in summary, in the first part, what we have done is that we have three networks. We have medicinal plants and their phytochemicals, that is our centerpiece. Subsequently, we have made a lot of analysis of these phytochemicals. We find that there's a large drug-like space, and also a large fraction of this drug-like space which is not similar to any of the approved drug. There is only a 25% overlap between our set of phytochemicals and phytochemicals in the Chinese herb database. So there is a unique space of phytochemicals potentially for Indian herbs. And finally, with this analysis, this suggests that the phytochemicals of Indian medicinal plants or medicinal plants in general are likely to be very specific pointers of human proteins. We also have a data set of medicinal plants and their therapeutic uses, and medicinal plants and their use in traditional Indian medicine formulations. But if you see, we have plant phytochemicals, we have plant therapeutic effect, but what we would like to do basically is to go from phytochemicals to target proteins to therapeutic effect. If we can link that, maybe that can basically give a mechanistic explanation of if potentially a plant or its plant extract could lead to therapeutic effect. So we expect that our platform, or we hope to provide a platform based on computational biology tools, which will aid in actually traditional Indian knowledge-based modern drug discovery. So with that, I would like to shift from the first piece of work to a second piece of work, from the potentially beneficial to potentially harmful, which is on endocrine disrupting chemicals. I'll try to speed up here because I won't have time for this extensive piece of work. But briefly, if you look at human body, roughly it has 100 trillion cells. And one of the challenges for the human body is to keep communicating between these different cells in the body. And broadly, at least, there are two systems through which the human cell communicates between these different cells. One, the nervous system. Second, the endocrine system. And this work concerns the endocrine system. If you look at the endocrine system, it is basically a system of chemical messengers or hormones enabling communication between different parts of the body. It basically consists of glands which secrete hormones into the circulatory system and thereby control distant organs in the body. So I have basically listed glands, hormones, and their effects in the body in this table. So the work concerns class of chemicals in our environment, which are known as endocrine disrupting chemicals or EDCs, which are defined by the World Health Organization as follows. An endocrine disruptor is an exogenous substance or a mixture that alters the functions of the endocrine system and consequently causes adverse effects in an intact organism or its progeny or subpopulations. These are dubbed as chemicals of emerging concern according to the World Health Organization. These EDCs are actually omnipresent in our environment. They are in air, soil, and water. Few of these EDCs are from natural sources, for example, in plant-based foods, while some of these EDCs are, of course, synthetic chemicals used in human man-made products, e.g. in pesticides, cosmetics, etc. So it is important to understand what is the mode of action by which these EDCs cause their effect. Basically, these EDCs interfere with the endocrine system in at least two different ways. They can either mimic the natural hormones and compete with them in binding to hormonal receptors, or they could indirectly change hormonal levels in our body by affecting the hormone metabolism. So if you are somehow able to disrupt the hormones secreted by these different glands, you can imagine that you can cause through these chemicals developmental disorders to fertility problems. So it is important to realize that there is a huge burden that the world is facing due to these EDCs. In the study in a Lancet Diabetes and Endocrinology last year, they have estimated that the disease burden, impact on healthcare, and loss of productivity due to EDCs is more than $300 billion in US annually and more than 160 billion euros in EU annually. So there was a report which was also commissioned last year and was published by the European Parliament, which found gaps in the definition of EDCs and the risk assessment. And the report actually led to a call by the European Union, which calls for a comprehensive framework to define EDCs and limit their use in various products used by the humankind in the next years. So actually this work which I'm briefly presenting is in the area of computational toxicology. If you look at toxicology, it is basically an international disciplinary field which studies adverse effects of chemical substances on living organisms. Traditionally, of course, toxicologists have used animal testing to determine the adverse effects, but due to ethical issues, animal testing and there are calls for alternative methods. So if you look at the EDCs, if you look at consumer products, there are at least 100,000 chemicals which are in commercial use, and most of them have not gone any toxicity testing. And computational toxicology is basically an emerging area which aims to build mathematical models to um, predict the adverse effects of these chemicals on living organisms. Um, Sorry for the disruption. Basically, computational toxicology is an emerging area which aims to build mathematical models to predict the adverse effects of chemicals on living organisms. So there are 100,000 chemicals, but if you look at US EPA, it has only tested about 8,000 chemicals to date. So we need mathematical models to predict the adverse effects of various chemicals. So our basic goal is to build network-based predictive models for these endocrine disrupting chemicals. But before we could build such models, we need highly curated data sets for EDCs. So one thing to remember is that if your model is based on trash data, the results of the predictions are going to be even more trash. So you need a highly curated data set. So there were basically three features we wanted in a highly curated data set for EDCs, which we started building at. One, we needed a supporting evidence in humans or rodents for these EDCs, because there are some resources which exist, but they combine information from fish studies to environmental monitoring studies or computational predictions. But we wanted a data set which is exclusively based on experimental evidence in humans or rodents. Secondly, we wanted to compile the adverse effect of EDCs, and there is hardly any resource. Actually, there's no resource to date which compiles the system level perturbations caused by these EDCs in humans, etc. Finally, what is even more important is to also compile the dosage levels at which these EDCs cause their adverse effects upon exposure. So these second and third feature were not there. With these short-term goals, we decided to design a 4-stage workflow to identify EDCs with supporting evidence in humans or rodents. So I won't go into the detail. Again, work for 3 years. I will completely skip this in lack of time. We started with about 18,000 articles, which could potentially contain information on EDCs. A couple of my students read all the abstracts, figured out there are 14,000 papers out of the 18,000 which could contain information on EDCs. In the second stage, we read the papers to see which papers could have evidence only for humans and rodents, led to 3,300 articles. Subsequently, we retrieved the chemicals which were tested for endocrine disruption in humans or rodents in at least one of these articles, and we arrived at 1,626 chemicals which were tested in at least one of the papers. This was still, if that sounded daunting, but that was not that daunting. But we went ahead was we wanted to basically evaluate these experiments, published experiments, to see if they had really evidence to our satisfaction to call them as EDCs. So I would like to mention this consensus statement by Soleki et al., which was published subsequent to a big conference organized by the German Federal Risk Institute in 2016, which was published here. They call for the weight of evidence evaluation for both adversity and mode of action to define these EDCs. So we evaluated that by reading each experiment and evaluating them. and I don't have details to show here due to lack of time. But we finally find that we find sufficient evidence for 686 chemicals, which we call as EDCs, based on 1796 published studies. Since we have the structure and all, we could compile diverse information on each of these chemicals. So a question which emerges whenever I present this work is that can't these be done automated in some machine learning approach? So this is an example of a very simple paper which we have curated. So as you see, my student picks the chemical, the system, the test concentrations, the observed adverse effects to a controlled vocabulary, reproductive perturbations, the dosage at which the lowest dosage at which the effects are observed, and so on. So and then we get a graph like this. So this requires really full text mining, understanding, interpreting the experiments. It cannot be done those so easily. So what we have finally is these 686 EDCs. We of course have classified them based on their environmental source, based on 7 broad categories, 48 subcategories. You see consumer products capture most of these EDCs. Second, we have a division of these or classification of these chemicals based on the type of the evidence we have. Is it only in vivo human, in vivo rodent, in vitro rodent, and such classification. Thirdly, we have done a chemical classification as well. We find that many of these are benzenoids basically. And fourthly, we have also uh, categorized the EDCs based on their adverse effects, which is one of the important things we wanted to do. For example, the effects observed have been classified from, for example, developmental, neurological to reproductive to endocrine mediated cancers. So what we see is that in our data set, most of these chemicals have reproductive effects. So they can cause fertility issues, for example. So, okay, this is all captured in a data set which is accessible online. But after building this, we have done some analysis, which I want to quickly summarize. One, we have analyzed, for example, list of what are chemicals reported by US EPA as safer chemicals, and also a set which is FDA inactive ingredients. These are, for example, colorants used in medicine, which are considered to be safe. What we find is that 10 chemicals in the safer list are EDCs, 44 are in the FDA ingredient lists are EDCs. We also analyzed a set of 1328 chemicals which are banned in EU and used for use in cosmetics. We find 101 of them are EDCs. So I will, in interest of time, quickly summarize by saying a couple of things. I don't have time to see this network analysis. We have also made a network of chemical similarity network of these EDCs. What we find is that there is a large cluster of EDCs which cluster together, and the color coding of each of these nodes, which is a EDC, is based on the number of benzene rings there are. So as you see, there is a large cluster which can be classified as follows, that they have at least one benzene ring, these chemicals. But there are also some isolated clusters, and there are also isolated nodes which are not chemically similar to any of these other EDCs. This shows that the space is highly diverse, and we can actually tease out some interesting features. For example, why this cluster doesn't cluster with this, although they are both containing a single benzene ring. We find that this is basically chlorinated benzene ring, this is brominated benzene ring. So similarly, we have also looked at the network of EDCs and the gene or protein targets, and we have teased out this information from in vitro assays, which are published by the US EPA ToxCast experiments, and we have been able to make a network for 383 EDCs. So basically, you join two EDCs if they share common protein targets using the metric called Jaccard coefficient, and the network looks like this. So if you see, uh, there is again a big large cluster. There are some isolated clusters and there are some isolated nodes. And here the nodes are the size of the node is based on the degree of it, and the two hubs are basically DDT, which is in pesticides, and octylphenol, which is a well-known carcinogen. But finally, we wanted to see if similarity of two chemicals in their structure leads to similarity in their targets proteins. And what we find is that there is hardly any correlation, and this actually projects a big challenge for the community uh, who wants to design predictive models for QSAR, or quantitative structure activity relationship, which rely on chemical structure similarity for making predictions. So I don't have time, but I wanted to say that we have also evaluated using our positive data set various ADMET predictive tools for toxicity, and we find their predictions is not good based on our data set. In summary, we have done this work. I talk about a harmful space and a chemical space. Both works have received a lot of, luckily, press coverage in international and television media. But in the end, I just want to thank the people who did the work. The three people who led the work on phytochemicals, Vivek, Karthik, and both Kartiks, and the people who Janani and BS Karthikeyan, who led the work on our endocrine disruptors. Both projects have received no funding, but my lab is well funded, and I'm grateful for that. I'm sorry I went over time, and if there is any time, if the chair permits, I will be happy to answer any questions. Thank you very much.